Now we're going to get into pedigrees, and I hope you find this easy, but just know there's some trickier problems that you're going to have to do related to pedigrees that we're going to get into here in a little bit. So a pedigree is just a family tree that indicates which individuals have a specific trait. Now, one of the first things that you need to do whenever you see a problem with the pedigree is you have to determine the inheritance pattern for the trait that's being mapped. So an example of inheritance pattern is the trait is autosomal dominant or it's autosomal recessive, sex-linked dominant, sex-linked recessive. I've really never seen pedigrees that are showing traits that show incomplete dominance or codominance. So you can expect that you're either going to be dealing with an autosomal or a sex-linked trait, or a trait that is dominant, or a trait that is a recessive trait. So when we're trying to figure out the inheritance pattern, the first decision to make is this trait, this disorder, this disease that's being mapped out, is it autosomal or is it sex-linked? If the trait is autosomal, then that means that the gene for it is located somewhere on chromosomes 1 through 22, which again were the autosomes. Or if the trait is sex-linked, it simply means there's a gene for it either on the X or the Y chromosome. Now here's your criteria for determining if the trait is autosomal or sex-linked. If males and females are equally showing the trait, we've talked about this before, then it's an autosomal trait. Now what this tells you is how to write your letters for the people in the pedigree. If it's an autosomal trait, then you're just going to write letters like this. If you determine that there's many more males than females that have it, and it's going to be a significant difference, they're not going to leave any question about whether it's sex-linked or not. You're going to see many more males than females having the trait. If you determine that it is sex-linked, then that's going to tell you you need to be writing your genotypes on the different sex chromosomes. So you're going to be writing genotypes like this. Now the second conclusion you're going to make is whether the trait is a dominant trait, which means that it's being coded for by a dominant allele, or is it because the individual has two recessive alleles? So is it a recessive trait? And again, here's your criteria for determining whether it's a dominant trait or a recessive trait. When you study the pedigree, if you do not see a skip generation, then that means that the trait is being coded for by a dominant allele. If you see anywhere a skip in a generation on the pedigree, then that tells you the trait is recessive. Now, we have a hard time sometimes recognizing skips. A skip is where you have two parents who do not have it, and they have a child that does have it. So it skipped a generation. Now, no matter if you're determining if the trait is a dominant trait or it's a recessive trait, this is telling you how to write the letters of the shaded individuals. So if we've determined that this is a recessive trait, you need to give the shaded individuals the lowercase letters. So this individual would be homozygous recessive. That is the reason why they have that trait. Over here, we determined that this trait is due to a dominant allele because we don't see any skip in the generations. So that means that you give the shaded a capital letter. So they have this trait because they have a dominant allele for it. So I'm just going to put an A, and I'm not going to add another one for now, and you're going to know why here in just a little bit. So again, to review, when you're determining whether the trait is dominant or recessive, if you determine it's dominant, you give the shaded a capital letter. If you determine it's recessive, you're going to give the shaded a recessive letter, actually two recessive letters, because there could be homozygous recessive for the trait. Now, once you determined why individuals are shaded in, either due to the recessive allele or the dominant allele, then you're going to go back and in the pedigree, you're going to write in the genotypes of everyone that is homozygous recessive. So you're going to go in and fill in every single person that is little a, little a. So let's practice on this example right here. Now, we know that people that are shaded in because they have that dominant allele. So that means the people who aren't shaded in are going to be the homozygous recessive individuals. So we're going to shade them all in. Now, the next part, step 3, says use those recessive individuals to help you determine the genotypes of those individuals that have the dominant trait that are shaded in. So to figure out what a person is, usually you can look at their offspring. If they don't have offspring, then try to look at the genotypes and of their parents, and that'll give you a clue. So if I come down here and I'm trying to figure out this person, and I know they have a little a child, I know one letter had to come from one parent, and that means that this parent had to have a little a to pass on as well. But they have to have a big A to have that trait. So that individual is heterozygous for the trait. Now let's figure out this last square here. This individual doesn't have any children, so look at the parents. The parents are going to give you a clue as to what the genotype of this individual could be. Mom can only pass on a little a. So that means that this individual had to have a little a from one parent and it has a big A from the other parent. Now let's do that exact same process over here in this one. So again, once you have it established who the little a individuals are, use them to help you figure out the other people in the pedigree. Now we know that one little a had to come from mom, and her other one would be a big A, because if she was little a, she would be shaded in. And the same thing for dad. We know that he would be big A, little a, because he had to have a little a to pass on to his son. Now let's take a look at sister right here. So in this situation, there's no children to look off of. So we have to go back to the parents. And the parents aren't going to give us enough information to actually answer what her genotype is. Her parents could have passed on a big A and a big A. Or maybe this parent passed on a big A and this one passed on a little a. So for this individual right here, we're not sure. And I like to put a question mark there. And that question mark simply means we do not know if they're big A, or big A, little a in this situation. So notice I just went back, and for the individuals with a dominant trait, we indicated they were big A, little a, or we have to put question mark, which means that they're big A, or big A, little a. You will never write for sure that someone is big A on a pedigree, because there's no way that we can be for certain that they're big A. They could have a big A, little a. It's just that we never see that little a trait develop because it's a rare allele. So we will never write big A on a pedigree. So keeping those ideas in mind, let's go through and let's figure out the genotypes for the individuals on this pedigree. So our first step is to figure out the inheritance pattern. So we're taking a look at the shaded individuals. And the first decision we're going to make is for the shaded individuals. Do we have an autosomal or a sex-linked trait? So we have to identify whether it's more males developing it or if it's an even number of males and females. And you can see that we have plenty of females that are shaded in. So that means that this is an autosomal trait. And what that means is we're just going to write letters like this. We're not going to put anything on the sex chromosomes. Our second decision that we're making about the people that are shaded in is whether this trait is dominant or recessive. So we need to look for some skip generations. We need to try to find two parents who are not shaded in with a child that is. And we can't find that one on this pedigree. So that means that this trait is autosomal dominant. So that means that the reason why this person is shaded in is because they have a capital letter. Now remember, step 2 says after you figure out the inheritance pattern, you need to go in and write the genotypes of everyone that is homozygous recessive. We do not write in the genotypes with the big letters first. We always write in the homozygous recessive ones. Well, if all these people that are shaded in have it because of a capital letter, then that means that the people who are not shaded in would be homozygous recessive for the trait. So let's go ahead and indicate all of the individuals that are little a. And now for that third step, we have to use them to help identify the genotypes of the other individuals. So let's start up here at the top with 1 and 2. Now again, you can either look at the offspring to help determine what their genotypes are going to be, or you can look at their parents. But in this case, we don't have parents for 1 and 2 on the pedigree. So we have to take a look at the kids. The fact that they had an offspring that was little a, means that they each had to have a little a. And their other letter is a big A, because if they are little a, then they would be shaded in. So 1 and 2 are both heterozygous for this trait. Let's go ahead and let's go over here to 3 and let's work our way across. Now, if you want for practice, you can go ahead and hit pause and you can come back and you can check your work here in a second. So this child right here indicates that 3 had to be big A, little a, because it had to have a little a to pass on. The same with this other individual. Now let's go down here to 4. 4 doesn't have any kids to use, so let's go back to the parents, and we can tell that mom can only pass on a little a. So this child has to have a little a, and its other a would be big. Again, if it was little a, then it would not be shaded in. Now when we take a look at this individual right here, it's going to be a question mark. There's no children to go off of. And then if we take a look at the parents, we know that the parents could pass on a big A combo or big A, little a. So this person is a question mark. They're either homozygous dominant for the trait or they're heterozygous for the trait. As a matter of fact, all of these individuals in here are also going to be a question mark. We will not be able to tell for sure unless they either have a parent that's homozygous recessive or they have a kid that's homozygous recessive. And you don't see that for any of these different individuals. This daughter right here is also going to be a question mark. Again, she could be big A or big A, little a from 1 and 2. And same with this final daughter over there. Big A or big A, little a. Now let's do another example. And again, if you want to hit pause, try to figure out the inheritance pattern, and then determine the genotypes of all the individuals in that pedigree. And I'm going to use A's. If you want to use A's, you can come back and check your answer here in a second. So whenever I take a look at this pedigree, I notice that there is more females than males. But again, if it's sex-linked, you're going to see way more males than females. So I know this is an autosomal trait, and I'm not going to use X and Y chromosomes for their genotypes. Now, the second thing I notice is that it's skipping a generation. You can see a skip right here. You have 2 parents who do not have the trait with a child that does. So that tells me that it is a recessive trait. Now again, whenever you decide the inheritance pattern, it always tells you the letters of the shaded. So let me go back, because up here for this one, we said that we had a trait that was autosomal dominant. So again, that's given us the letters of the shaded. They have it because they have a capital letter. They have a dominant allele for the trait. Down here we decided the trait is autosomal recessive. Again, that goes with the shaded. The inheritance pattern always goes with the shaded individual. So that means that this individual has a trait because they're homozygous recessive. So once we figure out the inheritance pattern and we know why individuals are shaded in, either because of dominant allele or recessive allele, then we are going to put in all the genotypes of individuals that are homozygous recessive for the trait. So we can go in and fill in all these people, little a, little a. We're going to use them to try to figure out the genotypes of the other individuals. So let's take a look at A. The fact that A has this child lets you know that this individual has a little a because they passed it on to the next generation. So they're going to be big A, little a. For B, B is a question mark. B doesn't have a little a child, and we do not know B's parents. So all we know is that he could be big A, and he could be passing on a big A with that genotype, or he could be big A, little a, and he has just not passed on that little a to the next generation. But he could have a 5th child, and if that 5th child is little a, then we would know for sure what B is. But with the information that we have right now, we can't determine, so he's a question mark. Now we do know all the kids, because mom can only pass on a little a. So all the kids would be heterozygous for this trait. Let's take a look at C and D. We can tell that C is big A, little a, because of this little a child, and so is D, because they each had to have a recessive allele to pass it on to the next generation. We can also figure out D's parents. They're going to be heterozygous because of this individual right here. But for F, F would be a question mark because F could get a big A from each parent, or a big A and the other parent passed on a little a. We won't be sure until we see the children, and if they have a little a, we would know for sure. Now, obviously, this next trait is a sex-linked trait because we can see that we have all males developing this characteristic. And we can also tell that's recessive because we have skipped generations. So here's two parents with a child that has it. Here's two parents with a child that has it, and so on. So there's plenty of skipped generations telling us that this trait is a recessive trait. Now, whenever you're doing a pedigree that's showing a sex-linked trait, first fill out the males because their genotypes are going to be very easy to determine. So since we figured out this was sex-linked recessive, and again, anytime we figure out an inheritance pattern, it's telling us how to write the letters of a shaded in individual. We know that this male is X, and we have to put a small letter to represent that it has a recessive allele, and that's why they have this trait. X little letter Y. And so the first males that you can always fill in are the ones that have the trait. Now, if you know the genotypes of the males that are shaded, then you automatically know the genotypes of the males that are not shaded. They're going to be X with that dominant allele, Y. So we can determine all of the genotypes of males that are unshaded. I'm not going to do them all to save time. Okay, with the females can be a little bit tricky. But there is definitely a female that you could determine her genotype. Any female that has the trait is going to be X little a, X little a. And if we look at this pedigree, you can see that we do not have any females that have the trait. Let me go back up here for the male and put X big A, Y before I forget. Now, another type of female that we can definitely determine is any moms of affected sons. So let's take a look at this boy right here, and there is his mother. And we know since this is sex-linked, this Y came from dad, and so that means that the son is inheriting his X from mom. So she had to have an X with a little a to pass it on to her son. Her other letter, though, would be that dominant allele, because if she was X little a, she would be shaded in. So again, she's a mom of a son who's affected, and they're always going to be heterozygous if they do not have this trait. So here's another son, here's mom, so she's going to be heterozygous. Here's a son, here's mom, she's going to be heterozygous. And we also know she would be heterozygous because she has a son that has it. Now, the third type of female that we can definitely determine is any daughters of an affected dad. They're going to be heterozygous as well. Now, when I look at this pedigree, it doesn't look like I have any daughters of a male that isn't already decided. So, for example, she's a daughter of a male that's affected. And we know that she gets one X from mom, and dad passes on his X. So that's why she would have an X with a little a, because that's all he has to pass on to the next generation. And that looks like that's the only female that is a daughter of a male that is affected. Now, for all the other females, they're going to be question marks. And what that means is we cannot determine for certain whether they're homozygous or heterozygous for this trait. So we have to put both, or we have to indicate with the question mark that they could either be X big A, or X big A, X little a. Now, I've put some more problems down here, and I'm not going to go through and label the genotypes of every individual, but let's just take a second to see if you can recognize the inheritance pattern, and then that next step as in identifying who's got the small letters and who's got the big letters, because that's the most important part when trying to read a pedigree. So when we look at this first one, I can tell that we have plenty of females that have it, and there's two males, so this would be an autosomal trait. And I do not see any skip generations. I do not have two parents that do not have it with a child that does. So that means that it is autosomal dominant. So again, the inheritance pattern that we determine always tells us the letters of the shaded. So that means the shaded people are going to have a capital letter giving them that trait. But they are not the individuals that we fill in first. So we always fill in our homozygous recessive individuals. So all these individuals here would be little a. All of the unshaded basically are homozygous recessive. So let's go ahead and indicate those. And then we're going to use them to help us determine the genotypes of the individuals that are shaded in with this dominant trait. Again, to figure out someone, if you take a look at their children, it usually gives you a key as to what their genotype is. And this individual had to have a little a to pass on. The other one comes from the other parent, but they have to have that capital A giving them that trait. Dad can only pass on a little a, so this son would be heterozygous. The male up here would be big A, little a, because he has a son that is little a, little a. This individual has a little a child, so they would be big A, little a. Dad can only pass on a little a, so this person would be big A, little a. So it looks like in this situation, all the ones with the trait are heterozygous for it. Let's go down to the next two. And this one, we can tell it's autosomal. There's plenty of females with the trait. I'm going to indicate autosomal. And now we need to look for some skips and look carefully. I almost missed it, but this one has a skip right here. You have two parents that don't have it with a child that does. So that indicates that this is a recessive trait. Again, this tells us the letters of the shaded. So all these individuals that are shaded in have two recessive alleles for the trait. Now we can use them to help us figure out the genotypes of some of the other individuals. And let's see if we can find a few individuals that would be a question mark in this case. We wouldn't be sure if they were big A or little a. And it looks like there would only be a few individuals we couldn't determine. We would know these parents were big A, little a because of this child right here. But if we take a look at the other children that they have, we know that they could pass on a big A, or big A, little a to this child and to this child. So here's a situation where we have a couple question marks. And then this last example, all males have this trait, so it's a sex-linked trait. And we can see that it's skipping generations like you see right here, so that means that it's recessive. And we would first identify the genotypes of the males that have that trait. They would have X with a little ay. Then we would go through and figure out all the males who don't have the trait. They would be X, big AY. And then as for the females, we know the genotypes of females that have it, but you do not have any females that have the trait. You would know every mother of an affected son. So this mother would be heterozygous because she gives the X to her son. Dad passes on the Y. Here's another mother of a son who has it. And here's another mother of a son who has it. And then we look at any daughters of affected males, but we've already got her labeled right there. So she's a daughter of an affected male. So she's going to get an X with a little a from her father. And then the rest of the females are going to be question marks. Again, X big A, or X big A, X little a. Next, we're going to look at some probability problems where we have to analyze pedigrees and use Punnett squares. Now, these can be very simple or they can be a little bit tricky. So let's take a look at example 1. It says, what is the probability that 6 and a spouse will have another child that can roll their tongue? When I look at the pedigree, I first determine the inheritance pattern. Since there's plenty of females that have the trait, it is an autosomal trait. And I cannot see any skips in any generation. So that means that this trait is being coded for by a dominant allele. So that's telling us the individuals that are shaded in have a capital letter, that dominant allele giving them this trait. So that means that all the other individuals that are not shaded in are homozygous recessive. So now I can analyze 6 and his spouse. So I can tell from this child right here that 6 is heterozygous for the trait. So now I can do this probability problem since I now know the genotypes of the parents. So it says 6 and his wife. So 6 is heterozygous. His wife is homozygous recessive. If I do the Punnett square, it says that the probability that they have another child that can roll their tongue is going to be 50%, or 1/2. So that's an example of a very simple probability problem. We knew the genotypes of both parents, and they're asking what is the probability of their next child having a specific trait. So let's take a look at some of these trickier probability problems. In some of these trickier problems, you might need to multiply the probability of two different events. So what I mean by that is, imagine if you are flipping a coin and you were asked what is the probability that you flip two heads in a row. Well, the probability of flipping one head is 1/2, and then the next time when you flip a coin, the probability of getting heads is 1/2 again. So you take 1/2 times 1/2, and then the probability of flipping two heads in a row would then be 1/4. So that is what I mean by you multiply the probability of two events. So for the first coin flip, it was 1/2. For the second coin flip, it was 1/2. We multiply those two together. So let's take a look at example 2, because that is going to ask you to multiply the probability of two different events. It says calculate the probability that the next two children of 6 and a spouse will have the trait. Well, the probability that they have this trait, tongue rolling, is 1/2 whenever they go to have the first child. And then when they go to have the second child, they have the exact same probability. It's going to be 1/2 as well. Just because they have a child that maybe rolls their tongue isn't going to influence the second child's ability to have that trait. So again, their probability of having that trait is 1/2, just like with the first child. So when we multiply these two events, that gives us a final probability of 1/4. Now, another type of tricky problem you might encounter is one that's going to ask you to factor in some known information to come up with this final probability. So example 3 says if 1 and 2 have a fifth child, so let's take a look at 1 and 2. And since they have this child right here, we know that 1 and 2 are heterozygous individuals. So let's continue on. It says, what is the probability that she is heterozygous for the trait? So anytime 1 and 2 have children, we see that these are the possible genotypes in their offspring. Now, it says that this fifth child that they do have is a tongue roller. This is known information. We have to factor this in. So we know that this child is not going to have that genotype. So out of the remaining genotypes that are left, the probability that they're going to be heterozygous is 2 out of 3. And that's the final answer. Taking a look at example 4. It says PKU is an inherited disease caused by an autosomal recessive allele. If two individuals who are carriers of PKU have two children, what is the probability that neither child will have PKU? So it says that their parents are carriers. So let's indicate their genotypes. Big A, little a would be carriers. So every time they have a child, the probability that their child is not going to have PKU is 3/4. So if they're having two children, let's first talk about child 1. That child has a 3/4 chance of being born without PKU. Child 2, again, has that exact same probability, 3 out of 4. And this is a situation where we're multiplying the probability of two separate events. And that gives us a final probability of 9 out of 16 that they're going to have two children and both of them will not have PKU. Example 5 says 3, 2 and her spouse have a second son. What is the probability he will have this genetic condition? And we see that in this pedigree here. And we can tell since we have males with the trait and only males, this must be a sex-linked trait and it's skipping generations, so it must be sex-linked recessive. So that means that these males that have the trait have a lowercase letter on their X. So we can go ahead and indicate those. And then we know since this son has the trait, he gets the Y from dad, he gets the X from mom, so she must be heterozygous. So let's indicate she's heterozygous. And let's indicate dad's genotype. He's going to be X with the big AY because he does not have the trait. Now, it says that these individuals have a son. So let's indicate that they have a son. It says, what is the probability that he will have this genetic condition like his brother? So let's set up this Punnett square. So let's cross mom and dad. So determining the offspring genotypes here. Okay. Now, some of you might jump to the conclusion that the probability that they have this son and he has this trait is going to be 1/4. But this is a situation where we have to factor in some known information. We know that they did not have a girl because it stated that they had a son. So we can cross through this information. So that leaves us with these two boxes left. And the probability that their son is going to have this trait is now 1/2. There's a 50-50% chance of having this trait. Example 6 says, what is the probability that 3-2 and her spouse could have another son that would have the genetic condition? So in this situation, it's not saying they do have a son that was born. It's saying that if they have another child, what is the chance that he would have this genetic condition? So we can just take a look at this Punnett square that we have up here, because again, those are the two parents that are being crossed. And if they have another child, what is the chance that they're going to have a son that has that trait? So in this case, it's just 1/4. We do not know if the next child is going to be male or female, so we have to factor that in as well. Example 7, what is the probability that 3-2 and her spouse could have 2 more sons with the condition? Again, it's not saying they have sons or that they have one and they're getting another son. It's just if they have 2 more children, what is the probability they're going to be sons with the condition? So the odds that the first child is going to be a boy with the condition is 1/4. The odds that the second child is going to be a boy with the condition is 1/4. So the odds of them having 2 boys, both with this condition, would be 1 in 16. Now example 8 says, what is the probability that A is homozygous for this trait? Now when I look at this and I see that there's just one male shaded in, I could jump to the conclusion that it's sex-linked, but I'm going to tell you, when they give you a pedigree that is showing you a sex-linked trait, it's going to be very obvious. You're going to have many individuals, and you're going to have a large number of squares, boys, shaded in with very few or no females. So it'll be very obvious if it's sex-linked. So since it's not super obvious here, it could go either way, then I'm going to say that we have an autosomal recessive trait. So that means this individual is going to be little a, little a. The question says, what is the probability that A is homozygous for the trait? So that would be this individual right here. So let's take a look at the parents. We know the parents are going to be heterozygous because they had this son. So when we set up this Punnett square, we have to factor in some additional information that we know before we can come up with this final probability. We know that A is not shaded in. So we know that this individual right here is not homozygous recessive for the trait. So what is the probability that this individual is homozygous for the trait? It would be this right here, which is 1/3, and that's our final probability. Now that I've shown you both simple and complex Punnett squares, you need to read carefully through the problem and see if this is a problem that's just going to ask you to do a simple Punnett square. We're not multiplying 2 different events, or we're not having to factor in known information. Or is it a trickier problem where you're going to have to do that, multiply 2 events or factor in some known information? When you are doing these last few examples here, one thing that I want to point out is this problem right here was just a simple probability problem that you saw even in your pre-AP class. You just had to set up the Punnett square between 2 parents and then figure out the probability that they had a son that has that condition, and it was 1/4. But since we were working some of these complex problems, then it was a little difficult to determine exactly what to do here because you didn't know if you had to multiply 2 events or if you had to factor in some additional information. So that's the hardest part to probability problems, is just identifying whether you have a very simple one or you have one that's a little bit tricky. So take your time and read your questions very carefully.